Welcome back to Genetics Part 2. As my first song introduction. Can't believe I'm going to put that out in the public sphere. Sometimes you got to take risks. It could be the next Cardi B, you never know. Okay, so we're picking up where we left off last time in our introduction to genetics and inheritance, followed by evolution. We've talked about different alleles, and let me just remind you that an allele is a specific version of a gene. So everybody has the same genes. Again, we're thinking about the human genome as like a blueprint for building a house, and a gene carries the instructions for a specific part of that house. Let's say like a bathroom, a master bedroom, an entranceway, a kitchen, and hopefully, most houses have those, right? So, in terms of people, we might be talking about blueprints for eye color, or hair texture, or blueprints for height, although it's much more complicated for height. But there's different versions, right? Everybody's eyes have some color, but those colors are going to be different, right? So just like most houses are going to have a master bedroom, but that master bedroom may look different, be arranged differently in space. Okay, so alleles are the different versions of genes. So how do you get different versions of genes? Well, you get different versions of genes through mutations, and there's many different types of mutations. A mutation is a change in the genetic code, and so there are mutations called SNPs, SNP, which stands for, oh boy, let me see if I can write it on here. Single nucleotide polymorphisms. Yeah, I won't finish that, but single nucleotide polymorphism. Oh, you can see it right here. I didn't have to write it. And so that is a change in one nucleotide at one nucleotide base, right? So here are two individuals, individual 1 on top and individual 2 here. And we all have two versions, like one chromosome, say chromosome number 2, because that's what they're talking about here. We have one copy from our mother and one copy from our father. Got my gum stuck in my teeth. And so that's why individual 1 has 2 copies, individual 2 has 2 copies. So if you go through the letters of the genetic code for all of these chromosome 2, you will see that the letters are pretty much the same, but here at this point that's highlighted in yellow, there's differences, right? There's a T, A. Remember, A and T always bond together, right? And C and G always bond together, right? So 3 of the chromosomes have CG, 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 and this one has TA. So at some point, there was a change, a mutation at one nucleotide base pair, okay? So that's a single nucleotide polymorphism. Those are probably the ones we'll talk about the most throughout the genetics part of the class. There's also things called microsatellites, which are sometimes also called STRs. STR stands for short tandem repeat. And those are areas where there's a certain sequence, like CAG, That's weird. This is written in a way that should be different, but that's okay. So where there will be repeated units of that, CAG, 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 and that number of repeats could be different from one person to another. Then there's also insertions and deletions, and they're pretty much what they sound like, where if you have a particular sequence and through some process another base pair gets added in there, that's an insertion, or multiple base pairs. It could be a whole chunk of DNA that gets inserted in there. Or there's deletions where a whole chunk gets taken out. And that can cause some issues. Those tend to cause greater issues than SNPs for the most part. And you'll see why in a little bit. But again, I just really want you to realize that genes, the word genes is not synonymous with alleles. Alleles are genes, but an allele is a particular version of a gene. Okay? So for any gene that has different versions, those different versions are called alleles. So here we're going to talk about how does DNA carry the instructions for proteins, and what does that have to do with sort of building a living being? Okay, so in the process of protein synthesis, creating proteins, there are two broad kind of phases that we talk about. One is called transcription, and the other is called translation. And there's a way to sort of remember the difference that I use that I'll talk about later if I remember. It breaks down to this. To transcribe something is to basically copy it down, right? Say like before copying machines, like handwriting it, just making a copy of something. And in this case, we're copying in a slightly different form. We are copying instructions from DNA to something called RNA. So DNA, remember, is usually housed in the nucleus in these long strands called made of chromatin, called chromatids. And these long strands can't leave the nucleus. All right, there are little, there's a membrane around the nucleus, and the little pores in that membrane are essentially too small, and the DNA is too large. So DNA can't leave the nucleus. And if you recall from looking at the picture of the cell in the first part of this lecture, the ribosomes, which are sort of the protein factories, are outside of the nucleus. So how do we get the instructions from DNA to the ribosomes to make proteins? Well, through the process of transcription, by which sections of the DNA instructions are copied into RNA. So RNA is ribonucleic acid. Recall that DNA is deoxyribonucleic acid. So RNA, there are some differences. One, you know that DNA is this helical kind of structure, right? And RNA is just single-stranded. Okay, so that's one difference. Another difference is that whereas DNA has A, C, T, and G for its nucleotide bases, RNA has A, C, U, and G. So there is no T. Okay. I don't know exactly why, but that's how it is, okay? So what happens during the process of transcription is that there's an enzyme that allows the DNA strand to separate, like you see here. So there's, it sort of unzips, okay? That enzyme is called RNA polymerase. I'm not going to ask you that, but and sort of free-floating, you see this little U here? These free-floating nucleotide bases are able to pair up with bases in the DNA, right? So whereas with DNA, A would bond to T because there's no T in RNA, A bonds to U, right? Recall, again, this is the DNA side, so that's why there's a T here, and it bonds with an A in the RNA, right? So the newly made RNA strand is here, okay? The bases are getting added here. So it copies essentially like one side of the genetic code, all right? So if you compare these RNA bases here to the DNA bases, you'll find that everything is the same except that where there would be a T in DNA, there would be a U in RNA. So C, A, T, C, C, A, T, T becomes C, A, U, C, C, A, U, and so on, okay? Now, you have, this may seem counterintuitive, but you have the exact same DNA in all of your cells. You have the same DNA in cells in your eye as you have in cells in your heart, as you have in cells in your toes, in your liver, and so on. So how do those, I mean, obviously all those parts of your body function differently. They're constructed differently. So how does that happen if the same DNA is in all of those cells? It happens through the process of DNA expression, which essentially means, so in all of those cells, you have the same blueprints. You have the blueprints for the entire house in all of those cells. But in the cells that make up the liver, the only sections of DNA that are really being expressed are those that have to do with functions of the liver. Okay, so when you're working on the master bedroom, you're not concerned about the kitchen, right? You carry the same blueprints with you, but you're only carrying out the instructions that have to do with the master bedroom, right? So the process of making these strands of RNA is expressing certain parts of the DNA. So when transcription is happening, it's not copying the entire genome. It's copying specific parts, okay, depending on what needs to happen in the cell at that time. So it's kind of like, think of your entire genome as like a library. Now, if you want to read a couple things about World War II or a specific battle in World War II, you're not going to attempt to carry all of the stacks out of the library into your home. That would be ridiculous. So that's like DNA leaving the nucleus. It doesn't happen. It can't happen. What you do is you take a specific book or group of books off the shelf, you check those out, and you take those home. So that's kind of like messenger RNA strands being created and leaving the nucleus. Let me just make sure. Okay. I'm not really going to ask you, I don't think, about this, but it's useful information because, the science of genetics has become more and more complicated. And there are these things called transcription factors. And transcription factors are specific proteins that activate genes, that repress the expression of genes, that upregulate, like turn up the volume on sort of certain genes, or that downregulate, turning down the volume on certain genes. So, there could be some little thing that binds to a certain receptor area, a transcription factor that either prevents the expression of that gene, or turns on the expression of that gene, or increases the expression of that gene. And oftentimes this happens in non-coding areas. So remember when I said that geneticists used to think, well, there's this junk DNA that doesn't code for proteins. It doesn't seem to do much, so we don't know what the function is. Well, some of those areas are receptors for transcription factors, for example. Okay, so you've got your little messenger RNA or mRNA. that's created in the process of transcription. Now, so let me just draw a cell, and you've got your nucleus. I'm just going to say nuke, and you've got your DNA, blah blah blah, and then you've got your little messenger RNA, right? mRNA. So that can travel outside the nucleus into the cytoplasm, and the messenger RNA goes to the ribosome. Okay, so that's what you see in this diagram here. I'll explain it, and then I'll do some fun little drawings on the next page because there's a bigger diagram. So the messenger RNA goes to the ribosomes. It's sort of fed through the ribosomes. And then there are these sort of free-floating transfer RNA molecules, tRNA, that's this here. And those carry groups of 3 nucleotide bases to sort of fit the puzzle of the messenger RNA nucleotides. So it's like a lock and key situation, right? So you'll see here, you've got GAG in that messenger RNA strand. The part that bonds with that is C, because C and G bond together, U, because there is no T in RNA, which bonds with A, and then C, which bonds with G, right? So GAG corresponds with CUC, right? And so this little transfer RNA molecule here also picks up amino acids, right? So amino acids are the building blocks of proteins, and these are amino acids down here. So you've got glycine, serine, glutamine, I think, valine, lysine. Don't quote me on that, but I think that's what there are. There are 20 different amino acids, okay? And basically, the instructions that are transcribed from DNA to messenger RNA and then fed through the ribosome are in groups of 3, and those groups of 3 are called codons. And the transfer RNA brings anticodons, and those anticodons carry these amino acids to create a protein. So let's look at something bigger, okay? So here you have a cell. Here you've got, this is the nucleus of the cell. In the nucleus of the cell, you can see a little part of DNA is being unzipped there, and a little strand of messenger RNA has been created. That messenger RNA leaves the nucleus, as you see, into the cytoplasm, messenger RNA, and it goes through the ribosome, where in groups of 3, it matches up with corresponding transfer RNA, and each of those transfer RNA molecules hooks up with an amino acid. Okay, so let's look at these groups of 3: GAG, GUG, AAA, UGC, UAU, GGU. Right? These are codons. Okay, and each of those codons. So let me just write this down. CODON. S. That's an S. It's really hard to write on this screen. Each of those codons matches up with anticodons carried by transfer RNA. So again, this is messenger RNA. And these are transfer RNA molecules over here. So GAG binds with CUC. GUG binds with CAC. AAA binds with UUU, and so on. Now let me just quiz you. What bonds with UAU? Think about it. I know I'm wasting precious video time. A, because the U would normally be a T. A in DNA bonds with T, but because this is an RNA, it's a U, and U, which would be a T in DNA, bonds with A. Okay? Now, each of these transfer RNA molecules also hooks up with a particular amino acid. Okay? So here we have lysine, valine, glutamine. Okay? And those amino acids will eventually, like you put I don't know, thousands of them, hundreds of them, depending on the protein together, and then you get a protein. And what makes a protein function the way it does is not just the series of amino acids, but it's a three-dimensional shape, based on the charges and attractions of the molecules. It forms itself into a three-dimensional shape that has certain properties. Okay. This, you won't really need to use this, but this is a chart where you're looking at codons, and you can see what amino acids correspond to those codons. So all the ones in the same color make the same amino acid. So this is leucine. All of these make leucine. And I think this is phenylalanine. This is serine. Okay, this is proline. All right. And what you'll notice is that multiple different codons can make the same amino acid. That's called redundancy. And that's a good thing. Because imagine that you have a CTG sequence in your DNA and there's a mutation, right? And all of a sudden, the C mutates to a T. Now you have T, T, G. Oh no. What if that screws up your protein and that screws up something else down the line, and that essentially gives you a genetic disease? Well, no worries, because you'll notice that CTG makes leucine and so does TTG. So that's good. So the redundancy actually helps to prevent certain mutations from being potentially harmful mutations, right? However, it can occasionally be disastrous, because these two are what are called stop codons, and this one is a stop codon. And a stop codon in the process of transcription, I believe. It might be translation too, but it says, okay, I think actually it is translation. It says, this is where you're going to stop making the protein. The protein stops here. So imagine that you have a TGC, which equals cysteine, CYS, and that mutates to a TGA, which is a stop codon. That would be bad, right? Because you're supposed to be making cysteine, and then maybe after that, you're supposed to be adding on more amino acids for the protein to grow, but instead you hit a stop codon and the protein is truncated. So that could be bad. I will post a video of this DNA recap separately. So that's kind of the general process through which DNA is, the instructions are carried out to create proteins, okay? So here are just some fun facts. There's about 3.2 billion nucleotide pairs in the human genome, but only 2% of that actually codes for proteins, right? So that's why scientists were puzzled originally. There's a lot of repeating sequences that are non-coding. That adds up to about 50% of the human genome. The average gene is about 300 BP. BP is base pairs, so nucleotide pairs. So the gene is only about 3,000 of those, right? So if you were to like A, C, T, G, T, T, C, A, blah, 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 there's like 3,000 of those in an average gene. There's about 25,000 genes in the human genome. And I'm not going to ask you, I don't think, about any of those things, but I will probably ask you about this at some point. That the sequence is about 99.9% the same in all people, which means that if you take any two people, in the globe, and you go through their whole genome sequence, A, A, T, C, G, G, A, C, G, G, C, G, etc., it's going to be the same 99.9% of the time. Now, within that 0.1%, though, because there are 3.2 billion nucleotide pairs, within that 1%, there are, now this number changes as we discover more SNPs, so this could be outdated, but at least 38 million SNPs that were discovered, as of a few years ago. Okay, so remember, SNP is single nucleotide polymorphism. So you'll notice that of these organisms that are listed here, humans have the largest genome size, but we have the same estimated genes as a laboratory mouse and as mustard weed. Interesting, right? And yet there's still so much variety that we are so different from those other organisms. Okay. All right. So now let's talk about inheritance. So you may have heard of Gregor Mendel in a high school biology class. Gregor Mendel was a monk in what's now the Czech Republic. At the time of his birth, it was part of the Austro-Hungarian Empire. And he drank beer all day. No, I'm kidding. What he did, instead of drink beer all day, like most sane monks would do, is he experimented with peas. They probably thought he was weird. So he did these experiments with pea plants between 1856 and 1863. And why was he experimenting with pea plants? Because he wanted to understand inheritance. No one really knew how inheritance worked. They knew that you shared some traits with your mom, you shared some traits with your dad, you look like a mix of the two. So the common thought was that there must be something like blending that happens. If your dad is tall and your mom is short, you'll be some average between them. If your mom has blonde hair and your dad has dark hair, you'll have like sort of a sandy, brownish colored hair. That's what they thought. But Gregor Mendel wanted to really kind of dig in and figure out the mechanisms of inheritance. And, well, instead of finding volunteers to have children with each other and, exploring a few generations of humans, which is, number one, unethical, number two, impractical, because it takes a super long time, to create a new human from a new human. He used pea plants because they have short generation times. There's not the ethical concerns that there are with humans, and they have many traits that seem to be binary. So where there's sort of like one option or another option. So, for example, green-colored peas versus yellow-colored peas, smooth-coated peas versus wrinkly-coated peas, and so on and so forth. So what he did first he sort of had to get a base level. He had to create strains of peas where he knew that if you cross, which means you sort of mate, you cause to reproduce, two green pea plants, that you would only get green pea plants. Okay, that's called a true breeding pea strain. Okay, and then he would do the same with yellow. He would cross a yellow with a yellow until he got all yellow. And he might have to experiment with that because that wasn't like a thing that all green pea plants did or all yellow pea plants did. Sometimes you would breed some yellow pea plants and get a green pea plant, and so he had to ensure that he had true breeding pea strains before he really started his experiment. So then he took individuals from those true breeding strains and he bred those together, and that's called the F0 generation. So this is a true breeding green pea plant, and this is a true breeding yellow pea plant. And he crossed those together, and what he found is in their offspring, which he called the F1 generation, there were only yellow pea plants. Only, that's not an O, yellow. Okay. Okay, so that was step one. Then he took two of those yellow pea plants from the F1 generation and he bred those together. And what he found is in the F2 generation, on average, you would get about 25% of those. 25% would be green, and then you would have 75% of those would be yellow. Now, these are simple experiments, and essentially really what Mendel did was, number one, he controlled the steps of the experiments very well, he documented them very well, and he did the statistics on them, and he was meticulous in a way that no one had been before. And so, from these experiments that he did on different traits of pea plants, he was able to come up with two what he called laws of inheritance. One is called the law of segregation. And so, you know, this is not segregation like in a legal sense, right? But segregation means to sort of separate things out. And so the law of segregation means that for any trait, for example, pea color, green or yellow, the offspring will inherit only one version of that trait from its mother and one version of that trait from its father. So if your mother had one green allele and one yellow allele, she could only pass on either green or yellow, right? And if your father had one green and one yellow, he could only pass on either one green or one yellow, okay? Then he also came up with the law of independent assortment, which means that if you take any two different traits, such as pea color and seed coat texture, so smooth or wrinkly, those are inherited independently of one another. So in other words, you could have a green smooth pea or you could have a green wrinkly pea. You could have a yellow smooth pea or you could have a yellow wrinkly pea, okay? So, you know, that's much like, for example, hair color and eye color. You know, you can have brown eyes and blonde hair, you can have blue eyes and blonde hair, you can have brown eyes and dark hair, or blue eyes and dark hair, green eyes, yada, yada, yada. Now, obviously, in that example, it's not the greatest example because you know that certain combinations may be more common than others, but that in no way disproves the law of independent assortment. Actually, when we learned later on about chromosomes, we learned that some areas are more likely to pass on together because of where they're located. So let's say here is a gene for eye color, and shortly thereafter is a gene for hair color. Recall that, you know, in the process of recombination, it's a random process. And throughout the whole length of the chromosome, the odds of the break happening in between these two exactly is the closer they are together, the smaller the odds are. So if two traits are located close to each other on a chromosome, they're more likely to get passed on together, right? But not always, okay? Now, there's another principle that we're all familiar with to some degree that he discovered, and that's the law of dominance, in which a dominant version of a trait will always mask a recessive version of a trait. Now, I want to be very clear that dominant does not mean better than or superior than. It has nothing to do with that. In fact, there are some dominant alleles that can be harmful. So it has nothing to do with superiority. It has nothing to do with being better for you or worse for you. So let's kind of put these together and take a look at what they look like in practice. So let's say we have our F0 generation. We're going to take those true breeding strains. So this is true breeding green, and this is true breeding yellow. And we're going to use the letter C, two versions of the letter C to describe our alleles. So we're going to say a capital C, a big C is for yellow, and it's dominant. We're going to say a little c is for green, and it's recessive. Okay, now, the law of segregation tells us that each of the offspring will inherit only one of the traits, or the versions of traits, from each parent. Now, in this case, each parent has two copies of the same allele, so it doesn't really matter. This parent can either pass on a little c or a little c. So 6 of one, half a dozen of the other. This one can only pass on a big C or a big C. So it should be no surprise that, whoa, hey. Okay, it should be no surprise that this one passed on a little c each time. So boom, 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 boom. And this one passed on a big C each time. Boom, 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 boom. So each of these in the F1 generation are big C, little c. So they carry one of each. All right. Now, let's take two of those F1 generation pea plants, the ones that have big C, little c, big C, little c, and let's figure out what kinds of combinations could be passed on. Well, this one could pass on a big C or a little c. So let's start with big C. Also, this one could pass on a big C or a little c. So our combinations, damn it. Our combinations are as follows. Big C, that's that. Big C, little c, big C, which we're just going to write as big C, little c. It doesn't matter the order in which you write them. Or little c, which is this, okay? I usually write them in the same order so that you can kind of easily see how many of the same outcome there are. Now, the law of dominance tells us that of those combinations, these three will all be yellow. And I know, yes, these two are the same, but there were two chances that could happen. So these are all going to be yellow because they all have at least one big C, and big C is dominant. So big C will always mask a recessive little c. And it tells us that only little c will be green. So that's what Mendel found, right? Only 25% would be green, and the rest would be yellow. So there's a name for these different combinations, so we don't have to just keep saying big C, little c all the time. The ones that have one of each, big and little, are called heterozygous. Hetero meaning different, right? The ones that have the same size letter are called homozygous. And if it's a recessive or a small letter, that's recessive. So homozygous recessive, and this, because it has two of the same size letter, that's also homozygous, but these are big C's, so that is homozygous dominant, okay? So you have heterozygous, homozygous recessive, and homozygous dominant. All right, so let's kind of put together all our vocab. The mechanism that controls the color of the peas is a gene. The different versions of that gene, i.e. big C and little c, are called alleles. A specific combination of alleles, like big C, little c, or heterozygous, is called its genotype, like the versions of genes it has. The physical manifestation of that, in this case, yellow peas, is called its phenotype. Phenotype, even though it's spelled with a PH, you can think pheno, physical, is sort of the physical expression of that. So, in this example, there are two distinct genotypes that result in the same phenotype. All right, we have heterozygous, and we have what? Homozygous dominant. Yeah. I'm assuming that's what you said, right? So those are the two genotypes. And the phenotype is having yellow peas. Okay. All right. Now, we're going to talk about the Punnett square here in just a minute. But first, recall that any trait like a yellow or green peas that's controlled by one gene or location on the genome and has a limited number of forms is called a Mendelian trait in honor of the beer-swilling pea breeding Gregor Mendel. All right, so green versus yellow peas. Blood type is actually a Mendelian trait. Whether or not you can drink milk as an adult without consequence, so lactase persistence or lactose tolerance, that's a Mendelian trait. Sickle cell disease is caused by a Mendelian trait. The vast majority of human traits are not Mendelian, but I'll get to that. So there was this British geneticist who came up with an easy graphical way to calculate the probabilities of different genotypes in the offspring of individuals when it pertains to Mendelian traits, right? And again, this is probably something you've seen before. So this is the guy, Reginald C. Punnett here. Seems like he should have a little curly mustache. So let's look at the first example that we did where, well, no, this is not the first example we did, but this is the first one we're going to do with Punnett squares. So we've got, we've got a yellow pea plant that is heterozygous, big C or little c, right? So based on the law of segregation, it can only pass on one of those at a time. So either big C or little c. And then we have a green one, which is homozygous recessive, so it can only pass on little c or little c, okay? So we separate those, right, around this 4-square thing, and then you just do your combinations. This combination, this combination, this combination, and this combination, okay? Now, so from that, we can say that these are all the possible combinations, right? And just looking at the probability, this happens 50% of the time, and this happens 50% of the time. So those are the genotype frequencies. The phenotype frequencies, well, this one, because there's a big C, and the big C is dominant, that's going to give you yellow peas, and the other one only has the recessive green, so that's going to give you green peas. So the phenotype frequencies are also 50% and 50%. But let's make it a little more complicated. Here we have two heterozygous ones, big C, little c, so big C or little c, big C or little c. And then we look at those combinations. Here you have big C, big C, big C, little c, little c, big C, little c, little c, okay? So the genotype frequencies are 25% homozygous dominant. That would be this one, 25%. Then we have here 2 out of 4, which is 50% heterozygous. And then we have here 25% homozygous recessive, which you see here. But if you look at the phenotype frequencies, 75%, 1, 2, 3 out of 4 are going to be yellow, and 25% are going to be green. Okay. Now that's on average. Not every pea plant has exactly 4 offspring, and not all the time do you have exactly 1 green, right? But on average, that's what you get. It's like flipping a coin. So As I said, a Mendelian trait is controlled by one or two genes that have a small number of alleles, right? So you can easily do those Punnett squares. You can easily calculate the probabilities of not only the genotypes, but the phenotypes. A polygenic trait is a trait controlled by multiple genes. Most human traits are polygenic, for example, height. And then there's something called pliotropy or pleiotropic effects. A pleiotropic effect is when one gene affects several different traits. So it's like a domino effect where maybe this one gene makes an enzyme, and that enzyme is needed in a variety of biological processes. So when we talked about biological sex and the various ways you can have intersex condition, one of them was 5-alpha reductase deficiency. So 5-alpha reductase is an enzyme, and an enzyme is just a protein. And that enzyme, 5-alpha reductase, is responsible for helping to break down testosterone into usable forms. So it breaks down testosterone into dihydrotestosterone and I don't know, something else. But if something goes wrong with that gene that controls 5-alpha reductase, that means that your testosterone can't be used efficiently by your body, and that will affect everything downstream. Everything that testosterone is needed for, it will affect. So that's a pleiotropic kind of situation. Just while I'm here, let's just quick talk about polygenic, and then I'll end this lecture. So controlled by multiple genes. Now, all right. So a Mendelian trait, like pea color, you've got yellow and you've got green, right? It's an either or situation, right? Now, there's basically going to be sort of, those are the only two traits, and maybe you have 75% yellow and 25% of individuals green or something like that. Okay, but a polygenic trait, genic, is one in which you don't just have two options. There's a distribution, right? And this would be like height. Now, we may colloquially say, he's short, or he's tall, or, something like that, but, or he's average. But we know that those are arbitrary distinctions. Those depend on the population and who else you're around. You might be the tallest in your class, but if you were on a professional basketball team, you would be like the tiniest person on that team, right? We know that people come in a variety of heights. Obviously, like extremely tall people are going to make up, the tail end of a distribution, and extremely short people are going to make up a tail end of the distribution. But there's all number of people in between, and there is an average. But that's going to depend on your population, right? So anytime you have a distribution of traits, that's not just like you have this gene or you don't have this gene, because that would result in a binary thing. There would only be people that are 6'5" and only people that are, 4'8". Okay, but we know that's not true. There's a whole spectrum in between. So that tells you that that's, most likely a polygenic trait, and in fact it is. Height is controlled by many genes, but not only those genes, an interaction between those genes and the environment, for example, childhood nutrition and health. So there you have it. Okay, so that wraps up this lecture. I will be back for the one on evolution. I'll see you soon. Take care.